Hello, myself Sourab, student of 6th semester BSc Botany honors Tufanganj Mahavidyalaya. Today I discuss about PCR and its application. PCR. PCR is a method widely used in molecular biology to rapidly make millions to billions of copies of a specific DNA sample. PCR was first invented by American biochemist Gary Mullis in the year of 1983. PCR methods amplify DNA fragments of between 0.1 and 10 kilobase in length, although some techniques allow to amplification of fragments up to 40 kbs. Now we talk about some basic of PCR. A DNA template that contains the DNA target region to amplify. A DNA polymerase, an enzyme that polymerizes new DNA strands. Heat-resistant Taq polymerase is especially common, as it is more likely to remain intact during the high temperature denaturation process. Two DNA primers that are complementary to the 3' ends of the sense and antisense strands of the DNA target. DNA polymerase can only bind to and elongate from a double-stranded region of DNA. A buffer solution provides a suitable chemical environment for optimum activity and stability of the DNA polymerase. To perform the PCR, the important thing for PCR is DNA polymerase, primers, DNA template, and nucleotides. Now we talk about primer design. First, primer length. It is generally accepted that the optimal length of PCR primers is 18 to 22 base pairs. This length is long enough for adequate specificity and short enough for primers to bind easily to the template at the annealing temperature. Second point is primer melting temperature, TM. Primer melting temperature by definition is the temperature at which one half of the DNA duplex will dissociate to become single-stranded and indicates the duplex stability. Primers with melting temperature in the range of 52 to 58 degrees centigrade generally produce the best results. Primers with melting temperature above 65 degrees have a tendency for secondary annealing. Now, primers should generally have the following properties to base result in PCR. Primers should be 18 to 24 bases, 40 to 60% guanine and cytosine content. Start and end with 1 or 2 guanine and cytosine pairs. Melting temperature of primer should be 50 to 60 degrees centigrade. Primer pairs should have a temperature within 5 degrees centigrade of each other. Primer pairs should not have complementary region. Now, DNA polymerase. DNA polymerase is an essential component of PCR due to its key role in synthesizing new DNA strands. Consequently, understanding the characteristics of the enzyme and the subsequent development of advanced DNA polymerase is critical for adapting the power of PCR for a wide range of biological applications. Third point, the use of Taq DNA polymerase in early PCR protocols. Significant improvements have been made specifically in the specificity, thermostability, fidelity, and possessivity of PCR enzymes. Now I discuss about the steps of PCR cycle. First step, denaturation. The reaction mixture is heated at a temperature between 80 to 80 degrees centigrade so that the dsDNA is denatured into DS means double-stranded DNA into single-stranded by disrupting the hydrogen bonds between complementary bases. Duration of this step should be 1 to 2 minutes. Second step, annealing. Temperature of reaction mixture is cooled into 45 to 80 degrees centigrade. Primer base pairs with the complementary sequence in the DNA. Hydrogen bonds reform. Annealing fancy word for renaturing. And the last steps and the third step, extension. Now the temperature is shifted into 72 degrees centigrade, which is ideal for polymerase. Primers are extended by joining the bases complementary to DNA strands. Elongation step continues where the polymerase adds DNTP from 5' to 3', reading the template from 3' to 5' side. Bases are added complementary to the template. Now first cycle is over and next cycle is continued. As per PCR machine is automated, third cycle of the same cycle is repeated up to 30 to 40 times. It is the diagram of PCR cycle with PCR machine. Now I discuss about types of PCR. Some common types of PCR are multiplex, nested, reverse transcriptase, real-time, touchdown, hot start colony, and inverse PCR. Multiplex PCR. Multiplex PCR is a variant of PCR method in which more than one target sequence is amplified using multiple sets of primers within a single PCR mixture. The technology was first used by Chamberlain et al. For the diagnosis of Duchenne muscular dystrophy in 1988. Nested PCR. Nested PCR is a modification of group design to increase the sensitivity and specificity of the assay reaction. It involves the use of two primer sets direct against the same target and two successive PCR reactions. RT-PCR, also known as reverse transcriptase PCR, is a variant of the polymer chain reaction that typically measures RNA expression levels. In RT-PCR, complementary DNA, cDNA, is made by reverse transcripting of the RNA template with the enzyme reverse transcriptase. Real-time polymerase erase chain reaction, that's it, real-time PCR, is commonly used to measure gene expression. Touchdown. TD-PCR is a modification of PCR in which the initial annealing temperature is higher than the optimal temperature mixing of the primers. It's gradually reduced over subsequent cycles until the mixing temperature or touchdown temperature is reached, much like the touchdown of an airplane. It is a diagram of touchdown PCR. Hot start PCR is a technique that inhibits hot start tag polymerase activity or the incorporation of modified DNTPs during reaction setup until a heat activation step occurs. Hot start PCR allows for reaction setup at room temperature without non-specific application in primer design formation. This diagram of hot start polymerase. Colony PCR is a rapid high throughput PCR method to determine the presence or absence of the inserted DNA into plasmid directly from the bacterial colonies. Molecular cloning is one of the most popular methods for DNA transformation since long. iPCR, also known as inverse polymerase chain reaction. In this method, it allows the application of DNA flanking a no known DNA sequence. The primers are facing outwards. Using the inverse PCR, the unknown sequence flanking down sequence can be readily amplified. Now I discuss about cloning of PCR. PCR cloning differs from traditional cloning in that the DNA fragment of inserted and even the vector can be amplified by the polymerase chain reaction and ligated together without the use of restriction enzymes. PCR cloning is a rapid method for cloning genes and is often used for projects that require higher throughput than traditional cloning methods can accommodate. It allows for the cloning DNA fragments that are not available in large amounts. Some advantages, advantage of cloning of PCR is high efficiency with dedicated vectors and amenable to high throughput. Some disadvantages of cloning of PCR: we have limited vector choices. It, it costs higher. Lack of sequence control at junction. Multi-fragment cloning is not straightforward. Directional cloning is difficult. Now I discuss about some application of PCR. PCR helps in modification of DNA fragments. Number 2. Diagnosis of pathogenic microorganism. PCR helps in DNA analysis of archaeological specimens. PCR helps in detection of mutation relevant for inherited disease. PCR helps in analysis of genetic markers for forensic application. Species to species amplification of DNA segments between interspersed repeat elements using primer based on sign sequence. PCR also helps in gene recombination. It is a diagram how PCR helps in gene recombination. Thank you.